So let's talk about allosteric control of our first enzyme, ATCase. We'll see that it is modulated allosterically through the binding of small organic molecules. ATCase is the abbreviation of the enzyme named aspartate transcarbamylase. This enzyme performs the first step in a series of steps to eventually synthesize pyrimidines. And what it does is it takes its substrates, carbamyl phosphate and aspartate, and converts them into the product N-carbamyl aspartate. And phosphate. What we know is that this enzyme is inhibited by CTP. CTP is pictured at the bottom right-hand side of the screen, but we should kind of recognize CTP because it is a nucleotide, and we've already learned about nucleotides in this class. CTP performs what we call feedback inhibition. This is when a molecule, spelling is important. Let's just try that again. Okay, so feedback inhibition is when a molecule made downstream, or a handful of steps away, comes back to inhibit an enzyme. What do I mean by this, okay? So here is the enzyme ATCase, right? Its product is called N-carbamyl aspartate. But this product, if you take it down through 6 additional enzymatic steps performed by different enzymes, will eventually become CTP. So CTP is a feedback inhibitor because it's made in the same pathway that this enzyme ATCase exists in, but it is not the product of ATCase. It is a product of the pathway. And we can see CTP performing inhibition if we look at some kinetic data here. We see that CTP pushed the graph to the right. And if we think back to kind of Km and Vmax ideas, let's say this is the Vmax kind of guesstimating based on the graph. Let's say this is 1/2 Vmax. We could identify the Km's for each of these curves. And you could see that, I know it's kind of hard to see. Maybe I should, let me just erase that and fix that. We can see how the Km for CTP appears to be larger than the Km for the curve without CTP at all. So we are seeing the inhibition happening. Now, why does it make sense that CTP would inhibit the enzyme ATCase? What is our conceptual understanding of why this should happen? Well, let's remember that ATCase makes let's go back to this slide real quick. ATCase is the enzyme, the first enzyme in the process that makes pyrimidine nitrogenous bases. CTP is a pyrimidine. So it makes sense. Oh man, spelling is rough today. It makes sense for CTP to inhibit this enzyme because the whole point of this enzyme is eventually to make pyrimidines, one of which is CTP. If we have enough CTP, do we need to make any more? No. So this type of feedback inhibition helps maintain appropriate levels of CTP. And once we've made enough, we don't need to make any more. So that's what's going on with CTP. But also from the graph, you may have noticed that it seemed like ATP had a smaller Km. So let's try this again. Vmax, 1/2 Vmax. So we'll see how the curve for ATP appears to have a smaller Km than the curve without ATP. So ATP is an activator of the enzyme ATCase. Well, recall again, folks, the point of this enzyme is to synthesize the pyrimidines. If we're synthesizing pyrimidines, we should probably keep in mind Chargaff's rule. Chargaff's rule basically tells us that the cell has equal amounts of purines and pyrimidines, right? Because we need the ATCG base pairing. So ATP is a purine. The base is a purine. So if we have high levels of ATP, we have high levels of purines. This is going to activate the enzyme ATCase to make more pyrimidines so that we can have an equal amount of purines and pyrimidines in the cells. So this makes sense to balance so we need to activate this enzyme if there's a lot of ATP around, because that is a signal that we have plenty of purines and we need to make enough pyrimidines. Excellent. So there's one last thing to mention here, folks. If you're looking very carefully at these kinetic curves, and I'll help us all out by erasing what's on this slide. If we're looking carefully at these curves, you might notice that the CTP curve looks a bit more S-shaped compared to the curve with no allosteric modulators and the one with ATP. So what's going on there? So for us to understand what's going on, to make that curve more S-shaped, we need to zoom out to the quaternary structure of this enzyme ATCase. So this enzyme is a dimer, right? Two subunits interacting with each other. What happens is when CTP binds, it pushes us from the R state to the T state. Let's remember that the R state means on or active, and the T state means off or inactive. We should understand that the CTP is pushing us from the R state to the T state. CTP is an inhibitor. But what do you notice is happening to the quaternary structure as we change to the T state? You notice how these two subunits are really close together. Now, if the curve is more S-shaped and you're seeing the subunits are coming closer together, we should understand that CTP is increasing cooperativity. An S-shaped curve tells us the S-shaped curve on this line tells us the enzyme is cooperative, okay? But how do we know that there's more cooperativity happening? Well, in this quaternary structure, when the subunits come close together and make those nice interactions with each other, they can kind of feel what's going on, right? So shape changes in one subunit would be more likely to impact the other subunit when they're interacting more. Let's compare that to ATP. Now, if ATP binds, we are pushing to the R state, which is the active state. However, there's going to be less cooperativity in the active site, because look how far away the subunits are now. So this is an interesting example of cooperativity because we're actually seeing more cooperativity by an inhibitor, which is confusing, but it all comes down to what are the interactions between the subunits. If the subunits are interacting more, we have more cooperativity. And that is our first example of allosteric modulation. Up next, glycogen phosphorylase.